So before I begin, let me remind you of our safe harbour statement. Cynata is a listed, publicly traded company, so please do refer to our filings on our website and on the Australian Stock Exchange platform for information about the company. A few background pieces of information for you. As I mentioned, we are listed in Australia on the Australian Exchange. We plan 3 phase 2 clinical programs to get underway in the next 12 months or so in graft-versus-host disease, critical limb ischemia and osteoarthritis, all with our proprietary Cymerus iPSC-derived MSCs. Technology originating from the University of Wisconsin, the home of FCDI, and also very much the home of stem cells. This technology is a novel, scalable, allogeneic MSC technology that utilises the enormous power of iPSCs. We recently completed a world-first clinical study, a phase 1 in GvHD with a highly successful outcome. The first time in the world that a clinical trial, as opposed to a clinical study, had been undertaken with an iPSC-derived cell therapy product. We have a strategic partnership with Fujifilm Corporation, who are an 8% shareholder of the company and who are our partner for GvHD. We have a strong balance sheet and well able to continue our program. We have a compelling preclinical data package with a range of preclinical studies having been successfully completed, and broad commercial potential of MSCs, which of course leads us to question why there is in fact so much interest in MSCs, and that interest is clearly illustrated on this slide with the cumulative number of MSC trials that have been commenced. The most recent data from clintrials.gov is around over 900 clinical studies underway around the world using MSCs derived primarily from tissue sources such as bone marrow or adipose tissue. Of course, in a forum like this, there needs little introduction to the therapeutic potential of MSCs, but very clearly, MSCs are potent mediators of regeneration of diseased and damaged tissue. They are very effective modulators of the immune system, and they clearly accelerate recovery of tissue from damage caused by disease or injury. And we're now finally starting to see the outcome of many years of intensive investigation with the approval in several markets of MSC-based products. In Japan, for example, with a product called Temcel, which was derived originally from the Osiris technology, and more recently, Alofacel from Tygenix here in Europe. Our own programs have been quite extensive. And on the right-hand side of this slide, you'll see a range of preclinical studies that we have conducted through our academic collaborators in a range of therapeutic targets that represent the typical investigative targets for therapeutic MSCs, such as coronary artery disease, diabetic wounds, pulmonary disease, and more recently, we have demonstrated very, very interesting utility of our MSCs in ameliorating the effects of cytokine release syndrome, CRS, or cytokine storm as it's often called. We'll hear a lot more about that, I'm sure, at this conference because it's associated typically with CAR T and other immunotherapies and can be a profound side effect, ultimately leading to death in patients who react very badly to those type of therapies. Our MSCs have shown very, very interesting utility in treating that condition. So we look to furthering our studies in relation to that. I mentioned earlier that we have planned 3 phase 2 clinical studies to get underway during the course of the next 12 months. In graft-versus-host disease, which is the subject of our option agreement with Fujifilm, in osteoarthritis, in one of the largest clinical studies ever undertaken with MSCs, and that's primarily funded by the Australian government's peak funding body, the NHMRC, and also, and unfortunately the slides messed up a little there, in critical limb ischemia. So a very broad range of applications for our technology, both preclinically as well as clinically. Turning now to the Cymerus technology, and quite simply, it utilises iPSCs and the ability of iPSCs to have essentially unlimited replicative potential as the power behind the technology. We all know that MSCs require very high doses of cells, typically 1 million cells per kilogram, originating from, say, a bone marrow aspirate, which yields around 20,000 MSCs per aspirate. So even for one dose, there has to be a very high level of population doubling to provide sufficient quantities of the MSC product. And of course, that becomes even more fundamentally limiting when we look to commercial applications that require clearly millions of doses. The power of iPSCs in being able to self-replicate provides us with the quantity of starting material that is so necessary to monetize or commercialize cell-based therapies using MSCs. So we expand the iPSCs and then ultimately in the final step of the process, we differentiate the MSC into the cell type of interest, which in our case, of course, is MSCs. Now, this is a typical protocol that many other companies are using today to generate their favourite tissue, and I'm sure we'll hear more from other speakers during the course of the next 2 days on their approach to manufacturing or utilising iPSCs to manufacture their cell therapy of interest. Then finally, what that allows us to do, of course, is to manufacture a highly consistent and reproducible product, which brings into stark contrast the current on-market solution to manufacturing MSCs, which, as I mentioned before, relies on multiple donors and massive expansion of the derived MSC product, which leads to two fundamental challenges. The first is the regulatory concern of different donor equals different product. Now, that is not yet played out in, for example, the FDA, and I'm sure we'll hear a lot more about that as time goes on, whether the FDA accepts multiple donors as being a consistent and viable commercial manufacturing solution. Moreover, what has become increasingly clear in the literature is that expansion of MSCs leads to replicative senescence, which ultimately leads to a reduction in potency, which may help explain why some of the later stage clinical studies utilising MSCs have been less than spectacular. Early studies, where only a small number of doses were required, have typically been very, very conclusive. But the later stage clinical trials, particularly with the failure of Prochymal in phase 3, the Cymerus MSC product, has led to a lot of investigation and an understanding of the replicative limitations of MSCs. Our technology, on the other hand, utilising iPSCs ensures we have, number one, consistency and reproducibility, and number two, that we maintain product efficacy, because the expansion that is required to produce product is undertaken at the level of the iPSC, not the MSC. So in essence, what we have is an evolution in technology, which we see on a daily basis in everything we see and do. And I've just used one example there. I mean, we all used to use a rotary phone many years ago, but now, of course, we carry a phone in our pocket that not only makes phone calls, but pretty much rules our lives. So technology led to the evolution of the telephone, microcircuits, battery technologies and so on. And we're seeing the same thing in cell therapies, where ultimately much more commercialisable, much more industrialisable manufacturing solutions will be developed. And our technology is one such solution, in our case, for MSCs. So turning now to our clinical study in graft-versus-host disease, which, as I mentioned before, had a highly successful outcome. The clinical trial protocol here, I won't dwell on it too much, but it was conducted in the United Kingdom following approval in that country via the MHRA. Many people question why we would go to the UK first. Simple answer is it was the quickest path to getting the product into the clinic and ultimately demonstrating whether the cells worked or not. We conducted the study in 15 patients with acute steroid-resistant GvHD. The clinical outlook for these patients is particularly grim. Depending on the study centre, ultimately within around 12 to 18 months, most of those patients are dead. So these patients have no further recourse beyond the failed steroid therapy. We had two dose groups, a high dose and a low dose, starting first with the latter. Both groups received 1 million cells per kilo or 2 million cells per kilo, high and low, 7 days apart, 2 doses. And then we undertook safety and efficacy assessments, which are summarised here in this slide. You can see that more than half of the patients demonstrated a complete response, that is, the complete absence of symptoms. So essentially the patients fully recovered, an outstanding outcome in this very difficult to treat patient population. Nearly all of the patients had at least an improvement of 1 GvHD grade, that was an overall response rate. And then, of course, most importantly, nearly all of the patients were still alive at the conclusion of the study evaluation period at 100 days. So again, a great outcome for these patients, their caregivers and their families, because as I mentioned before, the outlook for these patients is particularly grim. This slide also illustrates the individual patient data. So as you can see there at the start of the study, the little blue circles, coloured in circles, where the patients were enrolled in the study, and at the completion of the study at day 100, you can see with the triangles, all of the patients bar 2 improved. We had 1 patient die of pneumonia on the left-hand side there, and so was not available to follow up. Pneumonia is a common cause of death in patients with GvHD. And then the patient on the right, B5, was lost to follow-up. That patient decided to withdraw from the study after receiving the medication and go into palliative care. You've got to remember that these patients typically have undergone a long and arduous medical process in the course of having their bone marrow transplant, which has given rise to GvHD in the first place. And of course, as a prelude to that was the diagnosis of leukemia. So again, a very, very good outcome for this first in-human clinical study, with the trial meeting all endpoints, both in terms of safety as well as efficacy. This, of course, has enabled the company to move forward to a number of further clinical programs and, of course, to execute on our commercial model. We are a small Australian company, and so our goal is to partner the technology as many times as we possibly can to ensure that this platform is ultimately commercialised in as many therapeutic indications as possible. I mentioned our option relationship with Fujifilm in respect of GvHD, but we also are seeking partnerships for CLI and for osteoarthritis, and also for our preclinical programs in the areas that I mentioned before, including pulmonary disease and cardiovascular disease. The targets that we are pursuing in phase 2 do represent attractive commercial opportunities. CLI is a very challenging disease and growing worldwide, particularly in those demographics where patients just fail to take the advice of their physician and go on a diet or reduce their reliance upon alcohol and cigarettes and fail to exercise. It's about a $1.4 billion market for an MSC product. But osteoarthritis, of course, is the big one, and that's nearly a $12 billion market with a major health burden to our economies. Our phase 1 programs, a program in GvHD, of course, facilitated us getting moving on our phase 2 programs. Looking at the milestones coming up, as I mentioned, within the next 12 months, we plan to commence the 3 phase 2 programs, and then as our preclinical programs move forward and we continue to generate data from those that remain to be completed, we will use that as a showcase to attract potential partners for our technology. So in conclusion, we provide Australian Stock Exchange exposure to the growing regenerative medicine industry. We are the only company with a scalable manufacturing solution to MSCs that does not require multiple donors or massive expansion of the derived cell therapy product. We have an attractive partner-driven business model. All endpoints were achieved in our recently completed phase 1 clinical study in GvHD, demonstrating no adverse effects and highly encouraging efficacy. We have endorsement by Big Pharma with our relationship with Fujifilm, and a number of value inflection points will proceed as we continue to execute on our business strategy of licensing and partnering our technology. So again, I thank you for your attention and see that I'm just out of time, so look forward to talking with any of you who are interested in Cynata and our technology during the course of the next couple of days. Thanks again for your attention.